Welcome everybody, or welcome back to our next installment of our GHGA webinar. Today's topic is epigenetics with our guest Kathrin Kattler. Just to briefly remind you, or if you've never been here, GHGA stands for the German Human Genome-Phenome Archive. We're a consortium within the NFDI, and our mission is to enable GDPR-compliant human omics data sharing. So we want to enable the secondary use of clinical omics data in research to allow for more biological discovery, and also translation of biological discoveries into medicine. And that involves a lot of interactions between very diverse communities, like biologists, geneticists, clinical scientists, physicians, patients, etc., which is why we're offering these webinars to get all of these different groups together. And as I mentioned earlier, you can catch all of our videos on our YouTube channel. Nicole has already pasted the link into the chat if you want to check it out. And we also have all of the materials that we provide collected on our resources page under GHGA resources and training. And with that, I want to introduce our speaker today, Kathrin. She received her bachelor's and her master's of science in human and molecular biology, and then got her doctorate from the Department of Epigenetics at the University of Saarland, where she is now a postdoc in the Walter Group for Genetics and Epigenetics since 2022. And the Walter Group is also part of our GHGA consortium. And so with that, Kathrin, the stage is yours.
So then also from my side, welcome to today's webinar, The Beginner's Guide to Epigenetics. And yeah, since we have a lot to cover, let's dive right in. In part 2, we will talk about exploring the epigenetic landscape with a focus on epigenetic modifications and mechanisms. So in the human body, we have more than 200 or even 300 different cell types. All of them have their diverse and very specific functions, and you can already, by looking at these pictures, see that they have a very different morphology. So for instance, these neurons are very distinguishable from these hepatocytes, and their functions are completely different. But each of these cell types share the same DNA. So the genome of each cell in the body is the same. The DNA is the blueprint to generate proteins, which in the end then exhibit the specific functions of the different cell types. Now, how is it possible that based on the same blueprint, we can get cells which know how to do specific things? The answer to that is gene regulation. In the human genome, we have about 20,000 genes, but not all of them are active all the time. So for example, if we go back to the neurons and hepatocytes, genes which are important for specific neuronal functions will be active in the neurons, but they will be inactive in the hepatocytes and vice versa. Now, how is this regulated? Here, epigenetics comes into play. So the term epigenetics was already introduced in 1942 by Conrad H. Waddington as the branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. In a bit more modern terms, this means that in epigenetics, we study heritable phenotypic changes. Importantly, these changes are not mediated by changes in the DNA sequence, so the DNA sequence stays the same. Instead, they are mediated by reversible modifications of DNA and chromatin, which result then in altered gene activity. Okay, this can also be visualized by the famous epigenetic landscape, which was also termed by Waddington. So in mammalian development, we start with a single pluripotent stem cell. And depending on which of the differentiation trajectories the daughter cells choose during development, we will get different specific cell types with their specific active gene sets. And which genes are active? in each cell type is then regulated by the different epigenetic modifications. We have DNA methylation, non-coding regulatory RNAs, and histone modifications. And today I want to mostly talk about DNA methylation and histone modifications, but I will also say a few words at least on non-coding RNAs. But before we come to the specific different modifications, we have to think about how is it even possible that 2 meters of DNA can be packaged into this tiny nucleus in each cell. When we look here at this EM image, you can see something which looks a bit like pearls on a string. So the string is the DNA and the pearls are units which are called nucleosomes. Nucleosomes consist out of histone octameres. So histones are proteins. And we have 4 different variants. We have H2A, H2B, H3, and H4. They consist out of the core protein, and then they also have the histone tail, which protrudes from the nucleosome. This will become important in the next slides. Now the DNA is wrapped around this histone complex, and together DNA and histone form the nucleosome. And the nucleosomes are the core units of the chromatin. The chromatin, again, is the foundation of epigenetic control. As an example, let's look at this pancreatic cell, so the nucleus of a pancreatic cell. These cells are highly active, so there is a lot of transcription happening. A lot of messenger RNA is produced. And you can see here that the structure of the chromatin looks very light, so this is very loosely and not highly condensed chromatin. This means the chromatin or the DNA is accessible to transcription factors and also to the transcriptional machinery. So here genes can be transcribed. Another example is here the nucleus of an erythrocyte. Here you can see that most of the chromatin looks very dark. It's strongly condensed. So we call this heterochromatin. And here the nucleosomes are sitting very closely together, and the DNA is not accessible to transcription factors or the transcriptional machinery, so most of the genes are silenced and cannot be expressed. Okay, so now let's start with the histone modifications. As I already said, the nucleosome consists out of the histones and the DNA, and then the histone tails, which protrude from the nucleosome. And you can see here that there are a lot of amino acids which can be modified by methylation, phosphorylation, ubiquitinylation, or acetylation. And depending on which modification we have, and also importantly, which of the amino acids is modified, these histone modifications can have a very different effect on the state of the chromatin. So for example, if we have a trimethylation of lysine 4 of histone 3, then this is a modification which is often found at active promoters. So this is an activating modification. On the other hand, if we have a trimethylation at the lysine 9, so H3K9 trimethylation, this is a very dominant repressive mark, which is mostly found in large silence domains of the heterochromatin. The sum of all the different histone modifications is referred to as the histone code. And you can see that in euchromatin, so in the open accessible chromatin, we have a different set of typical histone modifications than in the heterochromatin. Okay, now we already move on to the DNA methylation. In mammals, DNA methylation is almost exclusively found at the 5th position of a cytosine ring in a CpG context. So in a cytosine which is neighbored by a G. DNA methylation is mediated by DNA methyltransferases, DNMTs, using S-adenosylmethionine, SAM, as a cofactor. And it results in 5-MC, 5-methylcytosine. Again, the methyl group at the 5th carbon of the cytosine ring. So then some important facts about DNA methylation. First of all, methylation of the cytosine does not change the DNA sequence. And it also does not influence base pairing. Methylated cytosine will still pair with a guanine. Moreover, this CG context is a palindromic sequence. So this means on the upper strand in 5' to 3' direction, we have CG, and we have the same on the lower strand. From here to here, it's also CG. And usually both the upper and lower strand are methylated at a given CpG position. 5-methylcytosine is more hydrophobic than unmethylated cytosine, and so it has an effect on the DNA structure and also on the stability of the chromatin. And very importantly, as an epigenetic modification, 5-methylcytosine is a reversible mark. Now, how can the DNA be demethylated? So there are two different pathways. The first, that was already discovered quite a while ago, is the passive DNA demethylation. During replication, we have on the daughter strand the inclusion of new, so the newly synthesized strand includes only unmethylated cytosines, here depicted in white. So after the replication, we have what we call hemimethylated DNA. The DNA methylation is only on the mother strand, and the daughter strand does not have this methylation. Now, the DNMT1, the DNA methyltransferase 1, which is also called the maintenance methyltransferase, comes into play. It recognizes hemimethylated DNA and then sets a methylation also on the daughter strand corresponding to the methylation on the mother strand. So like this, we can inherit the DNA methylation over replication. But in cases where there is no DNMT1 activity, we have a dilution effect over replications. So we will lose the information of DNA methylation. And this process is, for example, happening during early mammalian development. So since there's passive DNA methylation, there's also a pathway which is active DNA demethylation. And you can see this is a bit more complex. So active DNA demethylation relies on oxidation of the methyl group. So this is mediated by TET enzymes. And in the first step, 5-methylcytosine is converted or oxidized to hydroxymethylcytosine, and then further to formal cytosine and carboxycytosine. And both formal and carboxycytosine are a lot less stable than methylcytosine, and they can be recognized by TDT, which removes them from the genome. And we have here an abasic site where the nucleotide is missing, where the base of the nucleotide is missing. And this is then recognized by the base excision repair system. So a system of the cell which recognizes when the DNA is damaged. So when a base is missing, it reads the other strand, and then so there is a G, so it will insert a cytosine. This is then again unmethylated, and we are back at the start. Okay, so with the histone modifications, we've seen that depending on which modification we have, they have different effects on the chromatin. But with DNA methylation, the situation is a bit more complex because the effect of DNA methylation depends on where we look at in the genome. So in different regulatory elements, DNA methylation can have different effects. Importantly, CpGs are also not randomly distributed throughout the genome, but we have regions where their density is much higher. So for example, regions with a very high density are called CpG islands. And they're typically found in promoter regions of genes which are regulated by DNA methylation. In these CpG islands of active genes, the promoter regions or the CpG islands themselves are usually unmethylated. Then the transcription factors and polymerase and so on can bind, and the gene is actively transcribed. If a CpG island is fully methylated, as is shown here, then this gene is silenced, so it cannot be transcribed. But apart from the regulation on CpG islands, DNA methylation also has other regulatory functions. So for example, we can see here that in the gene body, especially in exons of actively transcribed genes, there's also DNA methylation. And here it can have a stabilizing effect on the transcription. So a completely different effect than what we have in the CpG island. In enhancers, the situation is much more diverse. So there we can have an effect of unmethylated cytosine, because transcription factors frequently like to bind better or have a higher binding affinity if the CpGs in enhancer are unmethylated. But there are also examples of transcription factors which preferentially bind or exclusively bind to methylated CpGs. So yeah, here the situation is quite complicated. And final feature I'd like to mention is that DNA methylation is also essential to silence repeats. So repetitive elements which should not start to replicate in the gene. Okay, with that, I would like to already come to non-coding regulatory RNAs. As the name already implies, these RNAs have a regulatory functions and they do not code for proteins. Examples for that are microRNAs. These are very small RNAs which regulate gene expression. They can do this, for example, by binding to complementary sequences in their target mRNAs, which can then lead to the degradation of these mRNAs, or also to the inhibition of their translation. So these microRNAs will downregulate the expression of these mRNAs. Another example are the circRNAs or circular RNAs. They have a so-called sponge effect on microRNAs. This means that they will bind to the mRNAs and keep them from binding to the mRNAs. So they will again upregulate their target mRNAs. And on top of that, they also have an effect on splicing regulation. And the last example I'd like to mention are the long non-coding RNAs. They have very diverse functions. Just a few examples. They can also have a sponge effect on the microRNAs. Again, here they bind them and keep them from doing their job. Or they can also have a direct effect on gene expression regulation or also on protein scaffolding. This means they are kind of like a framework for the for putting protein complexes together. So you can see that also non-coding regulatory RNAs have a very complex different functions. And of course, all of these epigenetic modifications interact with each other. So there is a very intricate interplay of all the different epigenetic layers, and this makes things quite complicated. So as a summary, we have DNA methylation, which is set by DNA methyltransferases, and passive and active demethylation, which is mediated by TET enzymes. Then we also have the different kinds of histone modifications with their diverse functions. And what I didn't mention so far, we also have chromatin remodeling complexes, which are regulatory proteins, transcription factors, and so on, and also the already mentioned non-coding RNAs. And all of these different things, they work together as the epigenetic machinery. So in this very complex system, if there's a failure on one of the sites, then this can of course lead to problems, and in a worst case, this can lead to diseases. Very prominent, for example, DNA methylation changes in cancer. We also have epigenetic contributions to the development of autoimmune disorders, of neural disorders, and so on. And epigenetics also plays a role during aging. So you see that it really makes sense to study epigenetics. And what's, at least I think, what is really the most interesting thing is to study different layers of epigenetic modifications together to get a better idea about the interplay of epigenetic modifications. So this slide already is kind of brings me almost to part 2 of this webinar, because here you see genome browser tracks of different assays, which we will talk about in part 2, where we can profile the different modifications that I presented on a genome-wide scale. And you can already see that in these different tracks, there are a lot of different signatures. And I hope that by the end of the webinar, we can all understand how we can get to such a picture. Okay, so I hope you got now interested into epigenetics, and if you want to do some further reading, I can recommend this book, Introduction to Epigenetics. It's quite new, and it's also open access as an ebook. Or if you rather like to browse the internet, then you might also have a look here at this website, which also provides further information. Okay, so with that, I'm already at the end of part 1. And I'm open for questions now.